Hello, I'm Ryan, and today I'll be presenting the 2017 discovery that ATGL-mediated lipolysis in brown adipose tissue is not essential for non-shivering thermogenesis as it is in cardiac muscle. To begin, a vertebrate's homeostasis demands that it maintains constant body temperature in response to changing temperatures of the environment. In response to that, an organism is going to generate heat by taking advantage of the electron transport chain and oxidative phosphorylation, even to the point of fetal cycling. And it's going to do so with a few proteins. One, uncoupling protein 1, of which the structure you'll see here. Uncoupling protein 1 stays in the inner mitochondrial membrane cells of BAT, brown adipose tissue, and UCP1 also counteracts the proton gradient and dissipates it, causing more energy to be required to generate ATP and more heat generated as a byproduct. PGC1 alpha also helps to activate PPAR alpha. In other words, it's a cofactor of PPAR alpha, which is essential for brown adipose tissue's transcription of key intermediates for substrate oxidation and heat production. So PGC1 alpha is also an essential protein. When we are exposed to cold temperatures, when our peripheral nervous system picks that up, our sympathetic nervous system will release norepinephrine, activating beta-adrenergic receptors, increasing cellular cyclic AMP, increasing protein kinase A, or activating them anyway, and then increasing the transcription of both UCP1 and PGC1A. Now, if an organism is exposed to decreased temperatures for long periods of time, cold acclimation of the sympathetic nervous system initiates increased count of adipose cells in brown adipose tissue, and hyperplasia and hypertrophy from accumulation of triacylglycerides. Now, this is important because triacylglycerides need to be broken down, and they will be broken down by ATGL, which is the main enzyme of this study, adipose triglyceride lipase, and HSL, hormone-sensitive lipase. Now, these hydrolyzed fatty acids are thought to activate UCP1 and serve as fuel for beta oxidation, i.e. heat, and use of reduced NADH and FADH2 for the electron transport chain and fetal cycling and more heat. This claim is backed up by the following findings. 1. Knocking out ATGL both in the whole body and in just brown adipose tissue could not perform, or at least mice that had that gene knocked out, could not perform non-shivering thermogenesis and displayed weakened ability to break down triacylglycerides stored in brown adipose tissue, causing hyperplasia and hypertrophy. Use of drugs to block ATGL-mediated resulted in decreased respiration, implying that there was less activity of the uncoupling protein 1 and fewer molecules for fuel. ATGL, when active, is necessary for PPAR alpha to serve transcriptional roles, and however, hormone-sensitive lipase knockout mice were not cold sensitive. It did suggest that ATGL is necessary for non-shivering thermogenesis. Now, if you look with me at table 1, you'll see the different types of mice used for this study. They used ATGL knockouts entirely in brown adipose tissue, in white adipose tissue, in cardiac muscle, and throughout the whole body. If you take a look with me at figure 1, you'll see that ATGL knocked out in the entire body, not just brown adipose tissue, causes hypothermia when exposed to the cold and hypertrophy in brown adipose tissue, though it doesn't affect the mitochondrial function of the brown adipose tissue. ATGL deficiency greatly reduced lipolysis in triglycerides as seen by figure 1, and also this shows that ATGL and HSL are key in both brown adipose tissue and white adipose tissue for triacylglyceride hydrolysis. Total DNA content studies were performed showing that without ATGL in brown adipose tissue or at all in the body, hyperplasia and hypertrophy occur because of buildup of triacylglycerides. Further, deficiency of ATGL was thought to be responsible for mitochondrial dysfunction in heart muscle cells. However, this data does show that these mitochondria were not damaged by electron microscopy photos. The cristae of the mitochondria are still intact. Moving along to figure 2, we can notice that non-shivering thermogenesis in response to acute cold exposure still occurs without ATGL in brown adipose tissue. However, the presence of this ATGL in brown adipose tissue is not essential for thermogenesis during acute cold. Inducible brown adipose tissue specific ATGL knockout mice, or the IBACO mice, revealed a remarkable decrease of the protein ATGL, explaining figure 2b which demonstrates lower glycerol in Ibeco mice due to lack of triacylglyceride hydrolysis, and figure 2c with lower triacylglyceride hydrolase activity in Ibeco mice as compared to the control. Figures 2D and 2E again confirm the hyperplasia resulting from the accumulation of triacylglycerides in brown adipose tissue as compared to control mice. However, figure 2F reveals that even the Ibeco mice could very efficiently maintain homeostatic body temperature in response to acute cold, confirmed by the large concentration of uncoupling protein 1 and oxygen consumption rates of the Ibeco mice, comparable to the control mice. If you look with me at figure 3, you can see that hydrolysis of triacylglycerides via ATGL in white adipose tissue is essential to non-shivering thermogenesis while in starvation mode at 5 degrees Celsius. In fact, figures 3A and C suggest that the knockout of ATGL in brown adipose alone did not cause hypothermia and maintained high levels of plasma fatty acids and triacylglycerols. However, figure 3B suggests that the knockout of ATGL in both tissues, brown and white adipose, did cause hypothermia. This suggests that white adipose tissue's ATGL lipolysis is essential for non-shivering thermogenesis. And also figure 3D did suggest that when ATGL was knocked out in brown and white adipose, fatty acids in plasma were low relative to the control. Moving along to figure 4, we can see that if there's insufficient ATGL in cardiac muscle and in effect the heart is not performing efficiently, non-shivering thermogenesis is impaired and hypothermia does occur. Figures 4A and D reveal that when ATGL is knocked out in heart muscle, Ibeco mice, body temperature decreased much more quickly at 10 weeks old. However, at 6 weeks old, the mice were more tolerant of the cold. This suggests that though greatly impaired ability to maintain non-shivering thermogenesis in AKO mice total ATGL knockout, and weaker or no ability to maintain non-shivering thermogenesis when ATGL is knocked out in the heart, or the Ibeco mice. Figures 4, B, C, E, and F provide greater detail into the AKO and Ibeco total ATGL knockout and heart-specific knockout mice with some ability to maintain non-shivering thermogenesis with increased left ventricular mass and decreased cardiac output. As mice grow from 6 to 10 weeks old. So as mice age, their heart function is decreasing. Further, this implies that the weakened cardiac function with age correlates with the cold intolerance of ATGL global knockout mice. The integration of the decreased cardiac function with age and the increased intolerance to cold with age implies that cardiac function is necessary for non-shivering thermogenesis. I'd also like to take a look at figure 5, which displays that mice which can't produce sufficient ATGL can adapt, survive, and even recruit normal brown adipose tissue. These tests were performed to see if ATGL deficient mice, ACO-CTG, where ATGL was only sufficient in the cardiac muscle, could adapt and survive in cold temperatures, to which our data suggests that they could tolerate the cold quite well. And since the total brown adipose tissue protein increased in the ACO-CTG mice, brown adipose tissue recruitment must have occurred, despite the lack of a significant difference between uncoupling protein 1 concentrations. Figure 6 displays that even in brown adipose tissue deficient of ATGL, non-shivering thermogenesis still occurs. Figure 6A and E suggests that global knockout mice could not escalate to metabolic rates of control, implying that the importance of ATGL-mediated lipolysis would have at least occurred in heart muscle. Figure 6B and F reveal that when ATGL was knocked out in cardiac muscle, metabolic rate was much lower when the beta receptors were agonized. Again, these beta receptors are going to downstream lead to the fetal cycling, which increases heat. Figure 6C and G suggest that when ATGL was knocked out in cardiac muscle, plasma fatty acids were also lower when the beta receptors were agonized, implying that more were hydrolyzed. Figure 6, D and H imply that ATGL of brown adipose tissue is not essential for non-shivering thermogenesis, since the ATGL knocked out in brown adipose tissue, those mice, Ibeco, still maintained high body temperatures when AACO, the mice with the ATGL knockout in both white and brown adipose tissue, at lowest temperatures, were at lowest temperatures in both cold acclimated mice and at neutrally temperature acclimated mice. This supports the theory that ATGL-mediated lipolysis in cardiac muscle is essential for non-shivering thermogenesis. in conclusion, I'd like to remind you that biochemists once understood that ATGL-mediated triglycerol lipolytic hydrolysis in brown adipose tissue was fundamentally indispensable for induction of non-shivering thermogenesis in response to cold. Thus makes sense if lipolysis is essential for 1, activation of uncoupling protein 1, the generation of reducing equivalents, and PPAR alpha signaling. The specificity of the location of this ATGL lipolytic hydrolysis was investigated in this study among 5 different mouse models to find that, contradicting the current view, ATGL activity in brown adipose tissue is not required for cold-induced non-shivering thermogenesis. ATGL is essential in white adipose tissue during fasting for non-shivering thermogenesis, and during exposure to cold, a healthy cardiovascular system is essential for non-shivering thermogenesis. And finally, with sufficient nutrition, non-shivering thermogenesis depends not on ATGL-mediated lipolytic hydrolysis in brown adipose tissue or white adipose tissue, but in cardiac muscle. However, without sufficient nutrition, white adipose ATGL-mediated lipolytic hydrolysis is necessary. Finally, the cardiovascular system is essential for healthy response to cold exposure. Thank you.